Yi, visiting us and giving a Terasaki seminar. Dr. Yi is really a pioneer in cell therapy, especially immunocell therapy for cancer. Dr. Yi currently is the director of MD Anderson's cell therapy for solid tumor. And also, Dr. Yi really kind of was previous at Fred Hutch Cancer Center in Washington, and then moved to MD Anderson 10 years ago, pioneered this field on adoptive cell therapy. What that means is using immune cells from the patient and basically stimulating, expanding, engineering the T cells that recognize that patient's tumor antigen and put back on the patient to eradicate tumor. So technology including TILs, tumor infiltrating lymphocyte, but especially ETCs, endogenous T cell therapy, where Dr. Yi actually taking blood cells from patient and using advanced algorithm to predict antigens from the tumor and stimulating, expanding the T cells to really fighting the tumor. He had quite a few landmark papers that really changed the field. Without further ado, I will invite Dr. Yi up to the podium and give us this very exciting talk. Thank you very much.
Thank you, Xiling. Wonderful to be here. I just came up from San Diego AACR meeting, a lot of exciting stuff. I think one of the things, though, that has been a recurring theme with cancer research is how do you tailor, individualize, personalized therapy? And I was going to call this talk something else, which is impersonal therapy, because you don't want to be too individualized in the treatment of your patient, or you won't have a consistent program of understanding the mechanisms of action and immune evasion. So a tumor agnostic patient-centric approach is actually, I think, something which is more informative, is able to democratize and move beyond some of the more challenging aspects of cancer therapy. So this, there we go. Here are my disclosures. I'm a founder of Mongoose Bio, typical founder of Maddox US, and a number of advisory boards, QR code if you want to use that. These are the people in my lab. This most important slide put at the beginning. I think that the two people who were instrumental in some of the research we've done in the last 5 or 6 years in antigen discovery is Kirpan on the left and Yulin Chu there, who's a computational biologist. Farah Hassan worked on tissue-resident memory cells. Yating Li and Jimei Wang worked on memory and exhaustion, and we'll get into some of that a little bit later. But down in San Diego, went spelunking in the ski caves, and I don't know if any of you know what this is, other than some edible shellfish. This is a keyhole limpet. So obviously, no immunologists here should begin their career without knowing what a keyhole limpet is, because the keyhole limpet produces something called keyhole limpet hemocyanin, or KLH, which is an adjuvant, which is still used in some cases treatment of superficial bladder cancer. But it was also happened to be Ted Geisel's birthday a few days ago. So I was on San Diego, this is the beach, and you all know who Dr. Seuss is, obviously. But this quote, "Step with care and great tact, and remember that life is a great balancing act," should apply to cancer therapy, especially immunotherapy, where there are both toxicities and efficacy, sometimes hand in hand, sometimes disparate, and then the ability of combinations to give meaningful responses. So I think one of the things through my career as I'm talking with Maurizio and Xilang and others is that you never know where you'll end up, right? So started in oncology, and I'm an oncologist by training. I do not have a PhD in immunology. I learned all my immunology on the job. And dabbled some in bioengineering, worked with actually some places up and down the coast here, Alba Medical, Berkeley Lights, and so on. Have been doing cell therapy for about 25 years, antigen discovery for the last 10 years, and then somehow wandered into space science, and we can talk about microgravity and T cells if you like. And of course, everybody works on SARS-CoV-2. So a little bit of that, some Parkinson's disease work, vaccine, and now we're hopefully revisit my favorite field of bioengineering. So here are the two basic questions. If you're an immunologist or a cell therapist or want to be one, is what makes a good target for T cells? How do we identify these targets? And then what makes a good T cell to adopt cellular therapy or even for vaccine therapy? How do we generate these cells, especially memory cells that are exhaustion resistant? And so since this is a tech-savvy group, we asked ChatGPT, what makes a good target for T cells? Someone asked me if I needed the sound. There's a typing sound actually that goes on here. But this was actually a pretty good response. I think this was the second iteration of this question, ChatGPT. This was 3.5, I think. But here are some features of a good T cell. One, foreignness. Okay, I'm not sure about that. I'm going to grade this paper in a minute. Abundance, meaning prevalence of expression of that peptide MHC or that T cell. Stability, that antigen doesn't go away. You know, once it sees something going on, it doesn't run off and hide. Immunogenicity is immunogenic. This is key. Immunogenicity is key. That's the only message I give you guys. Immunogenicity is key, because without immunogenicity, no matter what your prediction is, doesn't matter. It won't work. And finally, specificity. So let's grade this. Oops, sorry. Yes, they recognize peptides. That's true. An epitope, which is a peptide presented on the surface of tumor cell by self-MHC. Foreignness, no. Doesn't have to be foreign to be immunogenic. In fact, most of the immunogenic targets that lead to clinical responses are not foreign. Don't have to be ERVs or some fantastical dark genome splice variant. They could be, and they may be important, but they don't have to be. They don't have to be neoepitopes and neoantigens. They can be, but we think there's a little bit of overexuberance in that field. You kind of have to recenter yourself and say what is truly immunogenistic. Get that in a minute. And abundance, of course, is peptide MH density. If it's not expressed on the surface of sufficient density, you're not going to get a response. And this relates to immunogenicity, which is that peptide or epitope from that antigen you're interested in presented on the surface with sufficient peptide MH density to elicit a T cell response of sufficient affinity so that T cell or TCR recognizes the tumor target presenting that peptide MHC with sufficient density to elicit a response. So a bit iterative, but I think the other part is stability. And stability is essential to the persistence of a response if that epitope continues to be presented. And so we call that tumorigenicity. It doesn't have to be oncogene driver, but something that gives the tumor tumorigenic advantage is less likely to be lost. I think that when you talk about stability, you also have to talk about targets and the number of targets and whether multivalency is required. And there's a concept here called antigen spreading, which some of you may have heard. And that's important. We've cured patients targeting a single epitope to a single HLA allele with no other targets. And yet we're able to cure patients whose tumor variably expressed that target. So you think, you know, wow, you're going to have an angiloss variance. But that didn't occur because of the process of antigen spreading, where you can induce responses to non-targeted tumor-associated antigens. And that's an important concept to keep in mind as well. And there are ways to elicit antigen spreading, which are not being done right now, unfortunately, for a number of reasons, including high-dose lymphodepletion. And finally, specificity or selectivity, that the target is found selectively, doesn't have to be specifically, giving you some therapeutic window or some therapeutic advantage. You're treating that patient's cancer with a given T cell without doing a lot of damage to normal tissues. And so that's a pretty good, give it an A-, not bad. Okay, so first thing I'm going to talk about is epitope and peptide MHC density. So just to give you an idea how this works, you know, T cell sees a tumor and then it does its thing, right? Tumor looks bad, it's all blooding away, apoptotic, not good. And the reason for that is that, you know, the protein, which is a tumor antigen, is processed and presented, little bits of peptide end up on the surface. And then that's seen by the cognate T cell receptor, which then delivers a signal intrinsically and delivery of perforin, granzyme B, cytokines, what have you. And hopefully it'll do that and move on to another one, keep doing in a serial manner. Some of them don't even have to kill. But what's required in all cases is some type of receptor, a CAR T cell receptor or a TCR-T receptor or an endogenous receptor. And for CAR T cell therapy, you know, the receptor's been substituted by SFE region that binds a surface antigen. Remember, that receptor for CAR T cell has to recognize the surface antigen. The antigen, in almost all cases, must be present on the surface because it's not a peptide that's derived from a protein that's processed and presented. A TCR can recognize bits of protein from sourced from any compartment in the cell, whereas a CAR antibody receptor must recognize one on the surface. So that severely limits the number of targets available for CAR therapy. But TCR-T therapy, because the antigen can be come from anywhere, you can get multitude, tens of thousands of different epitopes that TCR can recognize. So what do we know about the status of TCR or CAR-T-based therapy? Well, unfortunately, really, this patient is smiling because he's got a target for which the epitope is known and for which you can give a T cell and take care of the tumor. That's really only 3% of the entire cancer patient population. If you take into account the target, the cancer type, the allele, everything, even CAR T cell. That's B cell malignancies. But for the other 97% of patients, we don't know what that target is. So how do we find these targets? And what do we know so far? This is a apple, core of the apple and the peel of the apple. The peel of the apple is surface antigen that's seen by CAR T cells. Surface accessible only, not HLA restricted, which is a good thing, but limited tumor specificity. Antigen loss is very likely. That's been seen in CD19 and even BCMA targeted therapy, responsible for a lot of the relapses that comprise 40 to 50% of those even achieving complete responses. So people don't talk about that very much, but the response rate is high initially, but the relapse rate is very, very high, 6 months to 2 years later. So currently available CAR targets, you've seen them here, B cell targets, some GD3, GD2, CEA, CD133, Claudin, you've heard a lot about 18.2, I think, in gastric cancer and other cancers. But the bulk of targets really come from the core of the apple, and that's the analogy. The way of the apple is all the proteins are available that can be processed and presented as bits of peptide on the surface. And they are HLA restricted, and that's actually a safety matter. But still, antigen loss is less likely because these proteins sometimes have a tumorigenic function, sometimes associated with proliferation or DNA repair and so on. But it's a shame, but really, there's no consideration in the industry right now for these tens of thousands of targets. They're focused on a few of them, MART1, GP100, the melanocyte-associated antigens, and a few cancer-testis antigens. That's it. There's not that much going on in the field because these are safe targets. They've been done before. Agents are available. Somebody needs to go through the difficult work of actually identifying additional targets and seeing, in fact, if they're immunogenic. And so let's focus on TCR targets. What makes a good, sorry, we did this already. I just want to point out that the key here is immunogenicity. And if you start with AI, not that it won't work, but if you start with it and you base your peptides solely on computation analysis, you do run into the problem of, is that peptide epitope actually presented? Does it elicit a T cell response? And does it elicit sufficient affinity to induce a tumor reactive, tumor rejection process? And then can you transfer that TCR? So unless you prove that and validate it, it's very difficult to say, I'm just going to do AI alone. We do the opposite. We do the empiric analysis first, and then we do AI. So for example, we would do mass spec, pull off the peptide, our surface MHC, and we have about 11 million spectra, all told, 6,500 targets. We deconvoluted that based upon ion score and other MHC binding and so on, but we already have the peptides. So applying AI afterwards still is empiric and allows us to produce a portfolio of targets which we think are potentially immunogenic. And now we prove that they're immunogenic by synthesizing that peptide, going through the pretty painful process of generating T cells that recognize the peptide, but not only that, also recognize tumor endogenously presenting that peptide of interest. So this is something not a lot of people do, or hardly anyone does. And if they do it from TIL cells. And the reason they do it from TIL cells is the presumption that there's a higher frequency of tumor reactive cells in the TIL biopsy, which is not inappropriate. However, your repertoire is very limited. A lot of cells don't grow and expand. We have been using peripheral blood as a source of these T cells that recognize peptide epitopes. And we've been fairly successful because the protocol we use, the ETC workflow, can isolate cells that are less than 1 in 1 million in frequency. You can't detect that clone type by PCR. That's how low they are. So we know they come from a very low frequency population. Gives us a fairly high success rate. About half of the epitopes we identified by mass spec turn out to be immunogenic, and we can make T cells against that. I'll tell you why that's important later. So yes, you can prove all these things, and then you can even transfer the TCR. This is a final nuts to bolts validation of the epitope you identified. So let's talk about a target that's been in the cancer research literature for some time called brachyury. Brachyury is a T-box transcription factor, essential for early embryonic development, but it's also associated with EMT, cell cycle progression. It's also known as TBXT. It is a pathognomonic diagnostic marker for the rare tumor chordoma, but it's also expressed in a lot of epithelial cancers. So as a gateway target, it's ideal. If you can target that and get breakthrough status because you're treating chordoma, but there's no reason why you also couldn't use that approach to target all these other epithelial cancers that share expression of brachyury. And so, as you can see in this diagram, a number of different approaches have been used using degrons or small molecules and so on to inhibit brachyury function. But we're not too interested in that. We don't care what the function is. We just want to make sure there's enough protein. And that's kind of interesting because some of these resistance mechanisms is upregulation of brachyury, which is exactly what we want. For T-cell therapy, we want more protein, so there's more peptide epitope on the surface. And so what's the story with brachyury? In 2007 and again 2014, an epitope was published describing the HLA-A2 restricted epitope purely based on MHC binding or net MHC criteria. And they generated these cells. We took them, we generated these cells, we stained them, both the wild type as well as the modified peptide. We sorted these out. Very nice population. You can see upper right-hand corner, CD8 positive, Touchman positive, W9V and W9L brachyury peptide epitopes. So kill the peptides super well, but they don't kill the tumor. And so this is about 7 or 8 different conditions and tumor types, and the middle two are interferon gamma treated or non-interferon gamma treated tumor, and the flanking ones are, sorry, the middle ones are the peptide and the flanking ones are the tumor, and then you see they don't kill tumor. So that peptide is either present very low density or is not process presented at all, and that is sort of one of the pitfalls of doing this all by net MHC, for example, and then there was a vaccine study which wasn't very successful, unfortunately. So we said, well, let's try and find in a very directed manner what that epitope is. So we overexpress brachyury in a cell line K562 that also expresses A2 or any other of the HL alleles that we were interested, run tandem mass spec on the peptides eluted through the tumor surface HC, and we got this huge brachyury peptidome, several tumor-associated brachyury derived epitopes that we now validated through the process that we described to you. So not only is the peptide there, I want to make sure it's immunogenic. So we went through this entire process, and we have 3 here. So I can't disclose them all. And we generated T cells, and you can see nice population here after 2 stimulations of brachyury peptide-specific T cells that we then sorted and expanded in uniform. Practically 90% of these are now peptide-specific for that brachyury peptide epitope. Do they kill tumor? Same question we asked before, and yes, they do. They kill, well, this is a peptide, first very high affinity. They kill very well, actually. 2 of them kill very well, 1 does not. But it turns out that these are very low E to T ratios. Most people use an E to T ratio of effector to target of 40 to 1 or 20 to 1. This is down to 2.5 to 1, 1/4 to 1. Killing tumor, in this case, chordoma and lung cancer, about 30, 40, 50%. So this is a true immunogenic epitope. We're pretty happy about that. So then, of course, SARS COVID epidemic came along, and everybody's working on SARS-CoV-2. So, okay, well, we got out of that. And so what's the story with SARS-CoV-2? Well, everybody focused on the spike protein because it's serologic response, give an antibody, blah, 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 but the spike protein is highly mutated. It turns out a number of high-profile publications came out, and if you add them all out, there was over 4,000 MHC class 1 and MHC class 2 peptides that were predicted. And they thought, okay, we got it, we got this immunogenicity, and among those, we got these 7 or 8 immunodominant peptide epitopes. We see it everywhere. Look at these responses, man. Even in patients who never got COVID, because maybe they were exposed to some other coronavirus, non-pathogenic like HKU or what have you. Major papers, major discussions. Only problem is they never actually checked to see if any of these peptides are immunogenic. So we took 1, 2, 3, 4, 5, 6, 7 of these, mostly from a spike protein, some from a membrane glycoprotein, all predicted, do they kill? No. Give me the answer right there. They don't kill. How do we know this? Because we went through the difficult work of synthesizing the peptide, making the peptide-specific T cells for each one of these. None of them kill. The only one that kills is number 8, and that was one we eluded. So what do we make of this? Well, we know that if you take a peptide that binds MHC well, even if it's a fake peptide or made-up peptide that binds those consensus genes, you will always get a T-cell reactive response, always. I can prove that to you. We've done it many times. In fact, we've done this assay with a positioning scanning combinatorial peptide library, put in an NY2-specific T-cell, and we came up with this very, very high indexed epitope, which doesn't exist in nature, but can generate NY2-specific T-cells, can generate random specific T-cells, irrelevant in terms of mediating a virus-specific or tumor-specific response. So we said, well, why don't we actually apply these first principles and find what the real peptides are? And so we did that. We took certain fragments of SARS-CoV-2, especially the non-structural protein region, the replicase gene, ran it through this protocol again, pulled up mass spec, deconvoluted the data, and we had a number of targets in all these regions as you see here. And interestingly enough, and not surprisingly, these non-structural protein epitopes are highly conserved among all the variants. And this keeps going, the latest variants, none of them are mutated and are potential targets for these T-cell NS-derived epitopes. Here it is, just showing the data. We generated them, uniform population, they kill SARS-CoV-2 positive. This was published a few years ago. So what's the conclusion from this part is that in cyclical prediction strategies may not achieve desired results. Now, it may be that among the 900 or so, or 4,000, there may be 100 that are truly eluded. But you're kind of mixed in with all that other irrelevant peptide. Immunogenicity is key. I think I've hammered that home several times. The current list of 4,000 may be all relevant. And a number of brachyury HLA-A2, A3, A24 epitopes have been defined by mass spec, extending the probability of using this actually as more of a pan-cancer antigen. So what did we find though? Let's go after the tumors, because that's what we're here to talk about, cancer-based therapies. We took about 38, a lot of tumors. I think there's 20, 30 different tumor lines across different histologies, some PDXs as well. Ran about 100 files. You can see 97 raw data files through the mass spec. Elin and Kay and Dr. McCoss and Dr. Wong were instrumental because we've done this before with 2 other mass spec operators and machines, and we get junk. Here, because Kay very, very carefully wrote the peptide off, preserved this, sent it to University of Washington, more than half of the ones we tested are truly immunogenic. We can generate T cells to recognize tumors. So pretty high hit rate. There were, I think, 11 million spectra, as I mentioned already, 6,500 targets. And what do we know about this? Interestingly, and probably not surprisingly, if it's a shared antigen, you can see the TPX2, it's shared across all these different tumor types. It's in black here, and you can see all the other ones here. These antigens have several unique epitopes that are shared, right? They're unique to that antigen. They're unique because they haven't been identified before. That's what I mean by unique. But that, if you share the antigen expression, you share that epitope for a given HLA across multiple tumor types. So we have a lot of internal validation that this mass spec works. And it's a little bit surprising, even lymphomas and leukemias will express the same epitope as long as they express the same antigen as some of the solid tumors as well. And since we did it in an unbiased fashion, you've got multiple HLA alleles, so not just HLA-A2, but Asian alleles, for example, cover more than 80% of the world's population. So how does this compare to what we know? I mentioned briefly on top, MAGE, the CT antigens, GP100, NY-ESO, MART1, the melanocyte-associated antigens. Their levels of expression in the respective tissues, like melanoma, is about 2.5 to 3 transcript per megabase, and even lower for some of the CT antigens. Yet they're immunogenic. The cutoff is around 2 to 4 transcript per megabase. This has been verified by the folks at Tübingen and Hans-Jörg Grauman's group and so on. If you have a TPM that's more than 4, almost for certain, it'll produce a protein immunogenic epitope. And then on the right-hand side, you see the sort of GTEx database, just to give you an idea what's levels of expression. Obviously, the CT antigen is highly expressed in testis. So what did we find? We ran the analysis, and we've got, this is a partial list, of more than 250. I see here 100 high value, but actually more than 250 targets which have been filtered for selective expression. Remember, these are tumor-associated non-mutated proteins, but they're highly immunogenic, and a lot of them are CT antigens. As you see, we filtered them for those which primarily expressed in testicular tissue and not other normal tissues. So CT antigen truly or CT antigen-like, and several epitopes for the same antigen, across different alleles, sometimes more than one epitope for a given allele, across multiple different tumor types. This is a pretty nice piece of data. And if you buy the fact that, even though they're not mutated, they are tumor selective. You can even look at tissue-specific antigens, like for prostate cancer. Well, you don't need the prostate. Really, it's an expandable organ, especially as you get older. And we got very highly expressed targets for those. So that would be considered tumor selective or almost tumor specific. And so what's immunogenicity of some of these targets? Now, we have all these targets to look at. Can they be easily lost? This is what we found. So we know the existing pool of peptides are derived from antigens which are tissue specific, like the melanocyte-associated antigens, or the cancer testis specific, like MAGE or PRAME, what have you. Okay, so that's nice. But that's all we have. Mostly A2. The ones we did find, we have a lot of CT antigens, like the one shown here. We also have one called HT1, which is code for CT46, which is HORMAD1. CT antigen's been around for about 10 years, 15 years. Nobody has an epitope for it. We have two epitopes, 1A2, 1A11. Tissue-specific, melanocyte, prostate, and breast cancer antigens, so essentially tumor selective. And then epitopes against cell cycle proteins, canonical complex proteins, DNA replication, DNA repair. And I find it astounding, but not everyone does that. You can generate T-cell responses, and T-cell responses do exist for cell cycle proteins and canonical complex proteins. You'd think they shouldn't exist at all, but they exist at extreme low frequency. You'll never find that until you have to go after the naive, very low frequency population to find these. And when you do, the tumors overexpress these, obviously. Very high therapeutic window between these and dividing cells, dividing normal cells. And they're really bona fide targets. So it's like a pan-cancer antigen. And then, of course, epitopes for resistance genes like alpha kinase A and MDR, and the multi-drug resistance genes across multiple alleles covering 80% of the patient population, very high levels of expression. Well, let's take a look at one of these, HORMAD1 CT antigen. HORMAD1 is very different from a lot of CT antigens. For one, it's expressed at high levels across multiple solid tumors, 40, 50% or high, multiple data sets for lung cancer, head and neck, esophageal, myeloid, bladder, cervical, and so on. It's a true CT antigen, only expressed in testis and not really in any other normal tissues. And then the interesting thing about this also is that it has very high H-score. The IHC has already been developed for HORMAD1 because of an interesting gene based upon its role in EMT transformation and DNA repair. So people are already looking to target HORMAD1 using small molecules and so on. So all the tools were there to give us a relatively high H-score. Other than NY-ESO for synovial sarcoma, this is one of the highest H-scores I've seen for CT antigens. And so HORMAD1 really represents a tumor-restricted, high prevalence antigen with tumorigenic potential. So it satisfies all the criteria that we'd like to see, generate the T cells, pretty high affinity, is peptide dose titration for the T cells itself, and they kill tumor fairly high. And this is an ET ratio of 10 to 1, 5 to 1, go at 10 to 30%. When you make the TCRT, not affinity matured. Its affinity is so high, actually, that we didn't even get to the tail end of it. We got down to 10 picomolar, and then it just dropped to 0. So probably even less than that. So much higher affinity than we would have expected. And then the killing down to 2.5 to 1.25 for the TCRT. It's not affinity matured, so not too worried about cross-reactivity. It's already been in vitro centrally deleted for cross-reactivity and number of disease indications, HLA-2 and HLA-11. And then, of course, we tested against all these different tumor types that kill really well. In the tumor model, expressing the lung cancer HORMAD-positive gene, it suppresses tumor growth very nicely. And then I don't think I'll go into this except to say that we've made a retroviral vector that's GMP grade, ready to run clinical trials with this. What about the other antigens here? We've got these other ones that I mentioned to you before that associate with cell cycle protein, canonical complex proteins, and found in a lot of high frequency tumors, but also pancreatic cancer. And you can see the CTL kills pancreatic cancer, but not GBM so much. But when you make the TCRT, they kill both fairly robustly. And then anaplastic thyroid cancer, which is sort of a local interest for us, with very high ATC population between here and also in Taiwan, and very high affinity killing, both with the endogenous T cell as well as the TCRT transduced PBMCs. What else have we got? We've got prostate cancer. I mentioned a few of these already, very high expression, found only in normal prostate tissue, also found in some other tissues as well, like gastric cancer. AML, it turns out very highly expressed, 166 TPM. This is actually a gametocyte-associated transcription factor, so only found in testis. Sorry, this one here, the second one here. And also T-cell and B-cell lymphomas. Some of these are neutrophil associated. Actually, the top one is neutrophil associated, so it's okay to transiently kill your neutrophils for a while, that'll recover. But also good for liver cancer across multiple alleles. Now we got breast cancer. I mentioned to you very high expression of mammoglobin, which is a biomarker for metastatic breast cancer. We got an epitope for that, very high levels found only in breast cancer tissue. And then there's another one, MYBR1, another CT antigen found only in breast cancer. But wait, there's more. What do you do about rare tumors? You can't run mass spec because it's hard to grow out rare tumors. So what do? You run the RNA-seq through our portfolio or immunopeptidome. If you got a hit, we got an epitope. You got an epitope, the HLA matches, we've got a target. And that's exactly what we did for glioblastoma, angiosarcoma, chordoma, and anaplastic thyroid cancer. Each one of these columns is an antigen target and an epitope. They kill very well. It spans across a lot of rare tumors, completely tumor agnostic, patient-centric. Here's an example of anaplastic thyroid cancer. We ran our peptidome across it, and sorry, I obliterated some of this, but the ones we can tell you are TPX2, TOP2A, PBK, and NDC80. PBK is actually highly tumor-specific, as it turns out. Some of these recognize TOP2A, topoisomerase 2 inhibitors, so treatment with that means that goes right up, makes an even better target. Again, they kill chordomas to varying degrees, but not the negative controls. And one thing that people don't do when they look at a epitope as an antigen TCR target is you have to actually throw in some cold peptide. Otherwise, you don't know if it's killing through the TCR. If you throw in cold peptide, you should decrease killing, so it distracts your T cell from killing the hot targets. And then you put in an irrelevant cold peptide, you should have no effect at all. So it's clearly killing through recognition of that peptide MHC. PBK, another target, we'll publish this soon, kills very well, as you can see. And then obviously HLA mismatch don't kill at all. What do we do with this? We cover a lot of rare and common tumors, cover a lot of the cancer patient population with all these alleles. We've empirically validated it all the way through TCRTs. And of course, all the applications you've heard about, either AACR or other places through industry, not only for endogenous T cell therapy, we use it to generate T cells from peripheral blood of patients, but also clone out of that TCR, use TCRT therapy, or put the TCR in NK cells or gamma delta cells, or your effector cell of choice. TCR mimics, bispecific antibodies, high affinity TCR bound to some other molecule like a drug, or obviously vaccines, which we've talked to you a little bit about today. And then finally, TCR coordinates response. This is really interesting because you got layer upon layer and layer of multiomics. You got single cell RNA-seq, you got single cell ATAC-seq, you got site-seq. What about the antigen-specific response? Right now, we need fresh cells to do tetramer staining, or fresh cells to do ELISpots. What if you could just decode the T cell receptor? So we work with Ling Wang and Guangxing Pei and Tao Wang over T Cell Western, who invented the PMT-net. They miraculously got all these samples from patients from China, single cell RNA-seq. So you get alpha beta chains out of these. And you also know what the expression level of that particular tumor and cell is that matches your T cell receptor. Even from a given patient, multiple samples from different sources, whether it's a primary tumor, whether it's the liver metastasis, whether it's peripheral blood. And now you can look at the clonal type alpha beta that are unequivocally paired. You can also look at the antigen expression for which we have epitopes to and see what lines up. We have 48,000 different clones. Turns out 418 are clonally expanded in the gastric cancer samples that we saw, and we just matched that up with our 252 epitopes and peptides, ran the PMT-net encode and decode algorithm, and let's see what matches. And so it turns out the more stringent you make it, the fewer you have, but hopefully of higher specificity, and this is the map that you get. Everything on the left-hand side is the TCR V beta sequence. We know the V alphas for them as well. Everything along the bottom was all these epitopes. You can see that a given T cell receptor recognizes a few, and we know the cross-reactor system recognizes a lot more. Let's get a closer look at this. So we look at something that's only increased in the primary tumor or in the liver metastasis, but not in the normal tissue, the same adjacent tissue. And you match it up and you see that they react to some of the TPX2 epitopes or a spindle protein epitope. So these are actually immunogenic, and they're naturally immunogenic, as you can see from these studies. So now we're going to deconvolute this and prove, in fact, that these TCRs are real, that if you know the TCR signature, you can now track, maybe just on FFPE stain, with the tumor-active T-cell responses. So you can apply this to therapeutics, immunodiagnostics, TCR correlates, and so on. This is a whole nother layer. And this is what I said to you before. If you target a single epitope, you may be able to induce antigen spreading. If you can induce antigen spreading, you get more antigen discovery. I'm not going to go into this in detail. I'm going to talk about it a little bit more later if you like. But the problem with current adoptive cell therapy studies is that everybody gets lymphodepleting conditioning. So how do you get antigen spreading? Not easy to get, and that is a problem. So I think, you know, there are a paucity of TCR targets for solid tumors. We have a way of defining it. We have a way of testing immunogenicity. I think we can translate these rapidly to clinical studies. We're doing that already. Let me spend the last little bit talking about what makes a good T cell. Now that you have targets, how do you find a good T cell? And one of the things that I like to describe a T cell is they have to be tenacious. What does tenacity mean? Ability to persist. They have to persist, and they have to be functionally persistent. Part of that involves clonal expansion. Part of that involves exhaustion resistance. I think it can provide long-term protection against a pathogen or a cancer cell, and can lead, as ChatGPT says, which I totally agree with therapies that are consistent with long-term protection. But these are things I talked about already. Okay, how do you generate this? What we do know is that there are TIL-based therapies and TCR CAR T therapies. And then there's something called endogenous T cell therapy, which Xiang mentioned already. Endogenous T cell therapy means you can isolate these T cells from the peripheral blood, and that makes a huge difference in terms of the longevity and the pre-existing replicative history of that cell. ETC really, I'm not going to go into detail how we discovered all this, but if you can isolate these from peripheral blood, you actually eliminate the requirement for high-dose lymphodepletion, high-dose IL-2, because we have a way using IL-21 priming and epigenetic modulation to turn them into memory cells. At the memory gels, and they're rare, you can use MES-based cell sorting to isolate and expand them to near uniform frequency of 10 billion cells per meter squared. Then you don't have to worry about all the other conditioning and high-dose IL-2 that's required. We've proven this in a number of studies. And this is the history since 1999, several first-in-human studies. One study using adoptively transferred CD4 T cell clones says you don't need anything at all. You don't even need low-dose lymphodepletion. These T cells persist and mediate complete tumor response. That was a New England Journal of Medicine paper targeting NY-ESO-1. And the interesting thing about that is NY-ESO-1 is heterogeneously expressed in tumor. So you expect outgrowth of NY-ESO-1 negative tumors, and that did not happen. Furthermore, most melanoma cells don't even express class 2. So that what was that CD4 T cell doing? It was recruiting effectors to the site, and we saw antigen spreading occur. But for CD8 T cells, if you add IL-21 prime T cells with anti-CTLA-4, you lower the threshold of activation, you do get antigen spreading. And so what's key in all these studies is that we showed that persistence correlates with response, antigen spreading here, we're targeting MART1, but you get responses to all these other non-targeted tumor-associated antigens, also correlates with response, leads to durable responses. But because there's a relatively non-toxic approach, no CRS, no ICANS, all those life-threatening complications, you can add combinations soon, you can add early, and so this is the new math. The math is this. If you use a broad TCR repertoire from your peripheral blood, you prime it to make memory cells, you get persistence. If you get persistence, you don't need conditioning IL-2. You don't need conditioning IL-2, you get antigen spreading because those endogenous tumor-reactive T cells are still there. And since it's not toxic, you can add combination therapy right away. Like the next day, you can add ipilimumab. So these two things add to a durable, complete response. What about memory? Memory can be characterized either as duration, as long as you can measure it, or the half-life. So very, very strong metrics. And we saw very early on, if we put in a clone that persisted, even though we didn't know anything about that clone, it turned out that that was a central memory cell. Even though they started looking like an infected memory cell, they revert to a central memory phenotype. This is random, stochastic selection of clone based upon proliferation work to grow out. So a clone, a single cell up to 10 billion requires 23 divisions. And Steve Rosenberg, when he saw this, said, oh, that's just a bunch of crap. You know, these cells are exhausted by that time. Well, he's wrong. because the number of doublings a T cell can have is 45 doublings. So if you get it at the 44th doubling, of course it's going to die. If you get it early on, it'll persist. That's how we found out that memory cells were important. But how do you make memory cells? And we found out that IL-21, if you prime with IL-21, you get a nice population of memory cells. You can see here, express high levels of CD28, and functionally they make their own IL-2 and divide very well, like memory cells should. So one of the, I think, most important studies showing that memory cells are important or critical to long-term protection is, let's give a patient, in this case, high-risk leukemia patient, an antigen-specific T cell that was not treated with a memory protocol. So these WT1 specific T cells was given to patients. This is an example of a patient. This is a T cell product, doesn't have any memory markers on it, has no CD28, has no CD127. And those T cells only persisted about a week long, and they didn't peter out, and they have no central memory markers. But if you give, this is an example of two different patients, the same T cell targeting specificity, but primed with IL-21, which you think induces memory, you get a nice central memory population to start with, and then at day 280, 9 months later, those T cells are still there. They have a central memory phenotype. Same for these two populations. And here you see relapses in all the patients who receive WT1 specific T cells without IL-21 priming. You can see that in black, the T cells persist and then they die, and the leukemia comes back. This is percent blast. For all the patients that received IL-21 prime WT1 specific T cells, they persist, leukemia never comes back. If we look at the half-life of these clonal types, for those who have complete responses, this is what you see in those two patients. Pretty high, actually, 50 to 150 days. Partial response, not so much. For patients with stable disease and progressive disease, the clonal type half-life was extremely short. But the half-life of your TCR clonal type that was antigen-specific that you treated patients does correlate with response. And not only that, if you take some of these that have been highly persistent and some that have been low persistent, even in the same patient product, you can then sort them into single-cell analysis, and you run up with a whole bunch of different populations, some of which are likely to be memory, long-term persistent, some which are not. The problem right now in the field is that not everybody likes to use naive peripheral blood cells. They like to just throw the TCR-T in the CAR-T. They won't go through the process of generating the T cells like with ETC therapy. So if you just throw in the TCR-T and CAR-T or use a TIL cell, they're mostly effector cells. And these are short-lived effector cells. How do you get them to be central memory cells? And we realized that, like I showed you before, effector cells don't last that long. Patients progress. Patients get central memory cells, the tumor goes away, and T cells persist. How do you get from one to the other? Well, it turns out all this is due to IL-21 prime phospho-STAT3 programming. And you can see here, if you start with a naive cell that hasn't differentiated, give them IL-21, phospho-STAT3 binds and you transcribe CD28. That's all good. But if you're an effector cell, you can't do that. It's already differentiated. Got a little chromatin here blocking phospho-STAT3 from binding to that promoter region. So you need to reprogram it. And if you do that by adding HDAC inhibitor, that gets knocked off. Then you can reprogram them. And now you get a nice population of central memory T cells here with TILs, CAR-T, and TCR-Ts. That's what we're doing. Taking this into the industrial arena with TCR-Ts. ETC, not so great. You have to do a lot of culture and stuff. But if you can reprogram them, then add the TCR, then you have a nice epigenetically stable population of central memory cells. I'll skip some of this. And how do you define exhaustion? Not only do they have to have high memory potential, they have to low exhaustion. And so working with Kunal Rai, who's an epigenetics guru, you see him right here in the picture, we looked at different cells heading towards central memory, heading towards effector, heading towards exhaustion here. And you can see that you can follow this as the trajectory moves actually from the left side here all the way through to the exhausted cells. And now you know what the master regulators are of exhaustion, knock those out, and you can mitigate progression to that. And we have several targets for this as a result of some of this. You can see trajectory moving towards exhaustion, not what we want. So final conclusion, high dose lymphodepletion, high dose IL-2 are really handicaps for T cell therapy right now. We don't want those. Too much toxicity, prevents antigen spreading, not good for adding combinations. Epigenetic programming yields true memory human CD8 T cells, proven that in clinical studies. And persistent causes of response, antigen spreading causes of response, and strategies to enhance persistence using single-cell omics can lead to translational targets. ETC does address several of these challenges, and then to move that into DCRT involves some additional programming. We have all this, we put it together in a program to address two gaps: new targets and TCR correlatives. We can drive this towards cell-based therapies or vaccine-based strategies and sort of program TCR-based therapy across a number of different programs at MD Anderson Cancer Center, a number of different tumor targets. And just rising from the immunopeptidome, you can give rise and spawn these additional projects. So last thing, because you guys may not remember any of that, something we all can relate to, beer margarita. So the beer is the effector cell. The margarita is what you put it in, right? So those who went to Korea, this is a pretty popular drink. Take the Corona, stick it in the margarita. Two great tastes last longer together. Basically, it really depends what your effector cell population is. And I put you here TH1 to 50, for example. CD4 T cells, you know, TH1, TH2, TH17, TH9. We know there are far more subtypes of TH cells. When we grew out the CD4 T cell clones, I say we had a CD4 T cell clone that made MCP1 and drove all the cells into the tissue. That's a great T cell to make because now you're attracting other effectors, causing more antigen spreading and a concerted response. So not just CD8 cells, but everything else must be considered, NK, NKT, gamma deltas. And you can genetically modify them, add some cytokines and chemokines, and then now add a checkpoint inhibitor, lower threshold of activation, do whatever you think is necessary, infiltration, agonists, which are now becoming more available, and then a whole different modalities to boost the response, to sustain the response, to induce tumor microenvironment violence, I guess, with oncolytic viruses, for example. All those things are good. The one thing about what we're doing, we think everyone should do, obviously, is take a very nice substrate of T cells where you know its specificity and phenotype, because then you can track it. Then becomes, you put it in a patient, it becomes a trackable cellular biomarker. So you can look at all these things, all your margaritas, to see what happens to that T cell. And you can interrogate it over time. And I think that's critical for moving the field forward. And that is it. Thanks so much for your time. Okay.
Would have tour de force. Probably a lot more immunology.
You guys just had lunch. You guys are falling, you know, it's a little postprandial.
Yeah, but that's great. I think that's kind of for the right people here. We have many engineers excited about this, you know, field, but that's kind of what you see is how advanced this feels right now from the textbook immunology you learned about. With that, maybe I will lead off a question. Wait, any question? Okay, all right.
Okay, I have three questions. The number one, like, what about HLA? Okay, what about HLA2 presented epitopes that can trigger help response for the stimulating or inhibiting cellular responses? HLA2.
Wait. So you said something about why only 3% or?
No. I am asking, what about HLA class 2?
Yes.
Class 2 presented epitopes.
Yes.
That can trigger or help the response for stimulating or inhibit cellular responses, like HLA2.
Okay.
It will trigger for cellular response?
Well, I don't think it matters whether it's HLA2 or not. Any class 1 MHC that has an epitope on it or a peptide should stimulate a T cell response.
So until now, like HLA1? Or?
So HLA class 1, so class 1 alleles, there's, I don't know, 40 or 50 of them. I think I showed in that table. The most common in the Caucasian population is A2 or O201. So everybody studies that one. But we're interested in all the other alleles as well, 2402, 0301, which are found in the Asian population. And a whole bunch of other ones, B40s and so on. So there's nothing, I don't think there's anything special about A2. It's just what we, you know, in Westernized civilization like to see more often. But I think that it's important to be able to address these other alleles as well. I hope that answers your question. Yeah.
So the another question, like, what do you think about interplay the NK cells?
Okay.
Yeah, like if it is recognized or not recognized. In that case, how about like HLA class 1 EFG?
Yeah, so that's good. So we're actually eluting peptides off HLA-E. to look at the NKT responses. I think the NK, the innate response, is important because obviously they're not HLA restricted. But even though people talk about NK memory cells, it's not clear that there's a defined NK memory path. Even the phenotype is still questioned. So a lot of times you'll hear people talking about NK cells being engineered to express a specific receptor. And I think that's not a bad idea, but you're still not going to recapitulate all the circuitry and machine required for T-cell memory. I think it's great. You may use it like a first wave, second wave. First wave is something that's readily available in innate cells. And it really depends on the stress molecules that are expressed by your tumor cell. Tumor cell is quiescent, you're not going to kill it with an NK cell. Not usually, I don't think. I think that, for example, we know that in pancreatic cancer, when they surveyed autoimmune population, gamma delta cells turned out to be correlated with response. Was that true unrelated? We don't know. But I think as we start to look at targets and we look at different effector cell populations, each one has a specific role. And even some of the gamma deltas in NK cells cross-present antigen, so they may actually serve as antigen-presenting cells instead of effector cells. We don't know. There's a study by Isaac Chen over at UT Southwestern showing different classes of NK cells. Some correlate with a good response, some negatively correlate with response, and you don't want to be growing those up, right? So those suppressor NK cells can be detrimental.
Thank you.
Okay. Any more on the list there? Yeah.
I have one more question. How about you think current immunotherapy or drugs triggering into this, kind of the pushing cancer, current immunotherapy and the drug,
So are you saying some pro-tumorigenic effect of the cells? Yeah. So that's a tricky question. I think some people believe that Th17 cells, so the IL-17 environment, is pro-tumorigenic. But it really depends on the timing and the context. If it's early on, it may be pro-tumorigenic, and some of that inflammatory process is actually detrimental to anti-tumor based therapies. So I think it's still up in the air whether Th17s are good or bad, or when you time it and when you give it. Certainly for toxicities, Th17 can sometimes lead to immune toxicities when you give immune checkpoint-based therapies. Yeah.
Thank you so much. It's a great presentation. Especially, I like the part IL-21. So I do have a question about trafficking of the T cells. One of the problem in the immunotherapy is that how the T cells is trafficking to the cancer cells. Do you have any something similar to the CCLX8, peptide similar to that one?
Yeah. By the time we're finished, we're going to cure cancer. So you guys asked all the really good questions. So how do you get the T cells to go where you want and stay there? One way is, we try to do is published in JCI and said, well, 3 years ago, was how do you generate tissue resident memory cells? They have some homing properties, CD103 positive, and so on. And of course, we're pretty arrogant. We think everyone else has been doing it wrong. And the reason why we think they were doing it wrong is they forgot to put in a little bit of hypoxia into the equation. And so as it turns out, if you take a naive population of cells and you subject it to the right hypoxic condition for the right duration of time during priming with TGF-beta, you will get a very nice population of tissue-resident memory cells, which are highly cytolytic, highly proliferative. Now, if you do the same thing, but you re-stimulate those cells under hypoxic conditions with TGF-beta, you get exhausted cells. So we're trying to study now, where's that threshold tipping one towards the other. Now, not all exhausted cells are bad either. There are precursor exhausted cells and there's whatever you, but I think we have to be careful about how we name these cells and whether that phenotype is stable. So, that was such a simple experiment that nobody's done, and it doesn't make sense. Why hasn't anybody done that? Because it's so much easier just to biopsy the samples and run your spatial transcriptomic study and say, here, we got this, we got that, we got this. It's not really, what should I say, choo-choo related all the time. But your question is, how do you get an infused T cell population? Let's say we don't have TRM, so let's say we even do have TRMs, but don't go where they want. They end up in the node instead of the tumor. When we gave the CD4 T cell clones, I think I just touched on real briefly. One of the patients we gave the clone to, and this was literally a clone of CD4 T cells recognized them. So you could see that the total white cell count dropped very quickly within the first 2 or 3 days and then recovered up 4 days later. Well, that's not marrow suppression, that's trafficking. And we did a white cell scan of that patient. You could see some of the areas light up. So that CD4 T cell, as it turned out, when we analyzed it in the lab, the cell product, very high levels of MCP1. Maybe that's one way to do it, right? The other ways are more intratumoral delivery, tumor-specific delivery, working with some guy at Texas A&M, bugs as drugs. You know, bugs will go to tumor sites. The perfect condition for bugs to grow. He's engineered them, taken all the pathogenic factors, and we put in something, and bang, you know, give a little bit of bacteria, get a little T cells, and off you go. So, you know, the common things, you know, inject some CCL21, inject some light, tag light to the tumor, you know, bispecific, what have you, all great ideas. I don't know what will work, won't work. But the one thing that I tried to point out was as you develop a persistent response, you might actually end up developing tertiary lymphoid structures. And those tertiary lymphoid structures are little factories for your T cells to resuscitate, go back to the tumor, back and forth like that. And actually, if you find those T cell structures in the tumor, then you're golden, right? Yeah, so lots of questions, but lots of tools also. Yeah. I'm surprised you guys are engineering backgrounds here. I was shocked. Yeah. Okay. Yeah. Okay. So, yeah, it's a big ask. Okay. So, I think first thing though, and we've started looking at this, is that we have true memory cells. They persist, and we have patients 10 years out, their cells are still there. That's great. You don't have to give lymphodepletion. You don't have relatively non-toxic. They stick around. Hopefully, they do their thing. So that's the first step, in a way, is to make those memory cells, put them in a patient. It could be tissue-resident memory cells, and hopefully they'll be protective for each patient. Now, that's autologous, right? You're not going to do that for everyone. There is a case to be made, and as you saw in the leukemia study, I'm not sure it was so clear in that slide, but those are patients with low tumor burden but high risk. Those are patients we love to treat with ETC therapy. You don't have to give high-dose lymphodepletion. Don't do anything toxic. Just get the leukapheresis product from them where the cells are relatively healthy still. Give them back to the patient and just sit back, and hopefully good things will happen. So I think that's one way to do it, right? Put in memory cells, low toxicity. Vaccines, obviously, and we talked about vaccines at lunch today, and we have so many targets for you guys if you want to put it in. And I think there's a really, really huge therapeutic window for T cells that recognize kinetical complexes and cell cycle proteins. You need just a tiny bit, or you need a lot, but it needs to always be there, and you can tune your TCR for that, or your vaccine, or what have you. And now you're preventing rapidly dividing cells from getting out of control. Well, those are usually your tumor cells. Some of them are very cancer specific. MDR genes, for example, or DNA repair genes, or CT antigens, or prostate specific tissue. You have vaccine against any prostate cancer cell, any prostate tissue coming back, you'll be cured, hopefully, right? Debulk, give the T cells, or give the vaccine, I mean, yeah. I'll repeat it, yeah, okay, yeah. Oh, yeah. So excited to answer that question. Hang on one second. sorry, so question was the high cost of goods, which is about $250,000 is cost of goods. Cost to the patient is $450,000. I know there's a pharmacist here. That's how much it costs. Cost to deliver the patient is another $200,000. Cost of maintaining, following the patient in the hospital, especially end up in the ICU with CRS, so about $1 million a shot. Okay, it's not 400,000. In the Anderson, it's much more than that. So first off, when we did this with the ETC therapy, did it practically in my lab. My GMP facility was a tiny little room, no bigger than your office, I'm assuming. And the cost of goods, even now, 10 years later, is less than $50,000. Okay, that's for 10 billion cells per meter squared. And to expand another infusion is $14,000. Why is it so expensive? One, because they can charge that much. Second is because you need a gigantic GMP facility to do all the gene vector manufacturing and the QA/QC, which is huge, and the processing, and you got to staff them, right? 7 days a week, whether you use that room or not. So we actually, at one point, tried to, well, actually, it wasn't me. Berkeley Lights, for those of you know what it is, it's a company that doesn't exist anymore, but they had a machine where you could use light to move the T cells or any cells around. And the guy, Kevin Chapman, called me and said, we want to put your ETC in a box. We want to take what you got, put it in a box. You don't need a GMP facility, you got a little box here, put in your leukapheresis product, and off you go. And they got really far with that until, for other reasons, the business climate made it difficult to proceed. But that's it, your point of care, GMP self-contained unit. And an attempt to do that at different stages was my working with Owl Bomb Medical in San Diego 15 years ago and helping them develop the nanosorter, MEMS grade cell sorter. The little valve flips back and forth about 50,000 times a second. Totally self-contained, isolate antigen-specific T cells, and then funnel that into something that expands them. So all this is feasible. You guys have all the technologies. I bet you have all the technologies here to do that. That would cost $5,000 a shot. Just in India, they're doing CAR T therapy. I think it's $20,000 or $30,000. Okay, so it's feasible. It's possible to do it. Partly because you don't have all the ramp up and everything. Everything is already there. But I think if you wanted to do this at point of care, it's perfectly feasible. People are talking about in vivo CAR T cell therapy. Like you have the vector, and the vector will automatically, insert itself into the right effector cell, and you don't even have to manufacture it. I don't know. Really, 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 really risky, I think. I think you have to have unequivocal targeting, probably a kill switch. Can't control anything about their epigenetics. Or I'm still enamored with getting a small number of cells. You put in 10 billion. I bet we could put in 100 million and have the same effect if we program those cells the right way. If you think about patients who get infectious mono, right? Gigantic lymph nodes, gigantic spleens from 2 or 3 clonal types. What the heck, That's incredible. We should be able to replicate that in some way, ex vivo. Hey, question, please.
Hello, my name is Menekşe. Thank you, Dr. Yi. It was a great talk. So I have a question, maybe a little bit branched, but anyway. So you presented the ways how you acquire the targets. So I will say it's a target acquisition, and you have different options. You can go for a pan-cancer target, or you can go for a cancer-specific target, or you can go even more in detail and go for like a patient-specific target. And then you talked about the endogenous T-cell therapies. So it is obviously, you can just like get your targets and you can train your endogenous T cells and just like inject it back to the patients. But when we look at from the target perspective, we see that like there are different paths with like, if you want to go for a pan-cancer target, probably it looks so different than a patient-specific target. So My question would be from a regulatory perspective or
Yeah.
from how you can make it happen
Right.
for the patients.
Right.
But I guess like for a pan-cancer target, we'll go full regulatory and like
Right.
make it as a drug. But for patient-specific, it should look completely different because it's like you cannot go for every single patient for sure.
Yeah, it's a really good question. So people have done things like they define targets, you know. Oh, so sorry. The question was, I hope everyone heard. You got different targets, different classes of targets. How do you decide, and what's a regulatory path for that? So you can make these, you know, red, yellow, green targets, right? And red ones really require super approval, and the green ones are truly tumor specific. And I would put CT antigens in the green category if they're true CT antigens. Safe bet, pretty immunogenic. And then the pan-cancer antigens in the red one, perhaps, most of them, not all of them, right? And then the tissue-specific ones, probably in the yellow category, prostate, breast, whatever it is. So your question is context, right? So the patient has lung cancer or breast cancer, well, there are multiple therapies available to them, even ovarian cancer, although that becomes worse with each treatment. You might have to gauge it according to that. But if you have a cancer of unknown primary, or you have a rare tumor, or you have leptomeningeal disease, then maybe all bets are off. Maybe you're going to take a look at that. The other thing we need to do a little more study on, and I think we already have the data for that that's available in the public domain, is these pan-cancer antigens. I mean, there is a pretty high therapeutic index there, right? We give chemotherapy, and we give target therapy, even kinase inhibitors. And we're talking, why are we holding T cells to such a, you know, incredibly high standard that it has to be truly tumor specific? I think there's enough clinical need that high therapeutic window will be fine for most people. I don't really believe in kill switches, so I might have to eat my words on that, but I think kill switches are good, maybe because you feel comfortable in your company and you want your stakeholders to feel comfortable. What do? You've already started the process, right? And, you know, if your targeting is so potentially volatile and toxic that you need a kill switch, maybe you shouldn't be targeting that antigen in the first place. So I don't know. I'm still kind of, I think that the other question obviously is the affinity of your T cell, the affinity maturation. We don't affinity mature TCR-T. But we do have a guy, I know this guy, so Tao has got this PMT-NET program, 3rd generation, where we're affinity maturing in silico and then testing it. And then I'm actually pretty happy with that because it seems to work very well with each iteration. And then you got some other people like Liz Wood at Jurabio, who's got this amazing AI technology. So I think we'll get to a point where I'll be very comfortable with affinity maturation and AI after we've decided what the target is in the epitope. So you're answering your question. Yeah.
Hi, Dr. Yi. It's great talk. I have 2 questions. One is that should we target for the CD4 peptide vaccine? So for the class 2 peptide could be better. Another question is that should we, for the mechanism research, should we target for the T cell differentiation, such as the T cell central memory T cell and the exhaustion T cell regulation?
Wait, what was the last thing you said? The last sentence?
Yeah. The such as the exhausting T cell.
The which T cell?
Exhausting. The T cell easily disappeared.
Exhausted T cell. Sorry. Okay, got it. Okay. So the CD4 T cell we like a lot. 2008 was when we published that study, the CD4 T cell clones. We just don't have an easy way of isolating and expanding them. And those tools are probably available now. And I think we should explore that a lot more. I know there was a study that I think Stan Riddell and Juno try to put CD4 CAR 19 and CD8 CAR 19 and put them together. That was such a painful study. In the end, they got nothing that was better. And you guys are really well read because you know what I'm talking about. And I think that not everyone knows this. Well, in theory, it sounded great, but in practice, it was terrible. And I think part of the reason was the selection was so crude. There was no other characterization. And, as close as monkeys are to man, they weren't. And it worked in CMV, but not in tumor-specific cells. So I would like to get back to CD4 cells. I think there are ways to tweak regulatory cells to become effective CD4 cells. And the tumor is full of regulatory cells, so why not take advantage of that? Put a TCR into a class 1 TCR into CD4 cell. You need some other stuff. You need CD8, you need a few other things. So it's a great opportunity to play with as long as you can be a little bit more careful about how you select the cells. Exhausted memory is a whole nother question. I don't know if I have time to go into that, but that's part of the epigenetic studies that we're doing, and we're targeting one of the master regulators right now, and hopefully that'll be effective. Yeah.
Yeah.
We got time. Okay. Okay.
Conclude this one.
Okay. All right. Thanks. Thank you. Great questions, you guys.
After that,